Guys, it's Mrs. Esser. I just wanted to quickly go over our enzymes, also known as our organic catalysts. The word catalyst and enzymes that can be used interchangeably. So looking at this first slide, we can see enzymes fall under our macromolecule of proteins. So we already know that enzymes are going to be considered a organic compound molecule. We know their building blocks will be the amino acid. Those amino acids will link together to form polypeptide chains, which will be folded into proteins. So we already know before even starting slide 1 that enzyme structure will equal their function. So just some enzyme basics. I always think of enzymes as a speeding up our body. So enzymes do not create energy, but rather they allow the same processes in our body to occur at a much cheaper rate. So I always think of our great show Zootopia, when they go to the DMV and we have the sloth who's moving so slowly just to stamp a pass for the bunny rabbit. Without our body, without enzymes in our body, think of it as our sloth moving very slow. So if there were no enzymes, chemicals would move way too slow for there to be life on Earth. So just some basic vocabulary before we get started. As I said, a catalyst and an enzyme, two words that can be used interchangeably. A catalyst is a substance that speeds up a chemical reaction without being changed itself. Key words, without being changed. So just from that sentence, I can see that catalysts or enzymes are reusable. They can be used over and over and over again, and they do not change from the reaction. Our third term is a substrate, is the reactant upon which an enzyme has its action. So substrate, reactant, two other words that can be used interchangeably. So think of a reactant in math. 1 + 1 = 2. 1 and 1 are going in, 2 is coming out. So substrate is the thing going into the reaction. And then a product, a substance that results from a chemical reaction. So what's what we are going to be ending with. Here's a picture of an enzyme substrate complex. So I already said enzymes are not changed by the reaction, only the substrate. So I can see this enzyme has a specific shape here at its active site. So think active site, it's where activity will be occurring. So the enzyme and substrate have the same shape, so they match up at the active site. And we have an enzyme substrate complex, aka where the enzyme and substrate meet. Our reaction occurs, in this example, it's going to be hydrolysis, so water would be added in order to split that substrate into our two products of A and B. So think of our enzymes as a lock and a key. Enzymes are very specific. They have one shape, as we saw in our last picture. This enzyme is only going to be able to act on substrates that have that exact same structure, okay? So enzymes like a lock and a key. If I use the key to my classroom for my car will not start because I need my car keys in order to open my car. Same thing, I need my classroom keys to open my classroom. So specific enzymes can only act upon certain substrates. Our example here, sucrase, will only break down sucrose. Protase only breaks down proteins. What do we think lipase breaks down? Probably lipids. DNA polymerase, remember polymer, many units. So DNA polymerase is going to build our DNA molecule. Lactase breaks down lactose. So if you are lactose intolerant, that means you are lacking the enzyme of lactase. Also, let's look at some other similarities between these words. Sucrase, protase, lipase, DNA polymerase, lactase, ASE. So our enzymes are going to be ending in that ASE. So if we see ASE at the end of the word, we can probably figure out that it's going to be an enzyme. So here's just looking at a rate of reaction. So as I said, enzymes are not energy molecules. They just make a reaction cheaper for us. So on the left, I can see the energy that would be needed in my body if I did not have enzymes, but thank you, yes, we do. So on the right, I can see this is how much energy my body needs with an enzyme. So I can see it lowered the amount of energy my body needs to supply in order to do the exact same thing. So putting it in different terms, let's say you wanted to buy an iPhone. You need to just save up for this iPhone, work many, many hours at work. And I came up to you and I offered you the exact same iPhone, but for 90% off. Obviously, you'd go with the 90% off instead of saving all of those hours to have that iPhone. So, our enzymes, they're like that discounted rate. Instead of paying 100% for the same product, you're only going to pay 10% of the energy. So, enzymes make our reactions cheaper and allow you to purchase your phone faster or allow you to break down or build things in your body faster. So, just some different factors that impact the rate of reaction, the concentration of a substance or a substrate, excuse me. So, if I have one substrate, it's going to be broken down, bada bing, bada boom, it's done. If I have 500 substrates and only one enzyme, my enzyme will work as fast as it possibly can on that substrate, but at some point, it's going to reach a point where it's at saturation, which means the enzyme's working as fast as it possibly can. pH stands for potential of hydrogen. We're going to be looking at the pH scale here in a moment. That's something that can cause the enzyme to denature. Remember, denature is when a protein falls apart. If our protein falls apart, it will no longer function. So it's very important to have our enzymes at the correct pH. Temperature is another thing that influences the rate of reaction or influences how the enzyme functions. So I can see warmer temperature will speed it up until a point. Once our enzyme gets too hot, it denatures, falls apart, and will no longer work. This is why when you're sick, if you get a fever of above 104 degrees, they recommend going to the hospital because if not, the enzymes in your bodies can begin to denature, which would be very, very bad for your health. At colder temperatures, the enzyme substrate reactions slow down just because they won't be colliding as much. Same thing with our atoms. When it's cold, they won't be hitting each other as much either. So I said I'd mention our pH and buffers just in case you weren't aware of those coming into our enzyme unit. So pH is the measured amount of hydrogen in a solution. As the number of hydrogen goes up, the pH goes down. So acids 1 through 6, thinking of lemon, stomach acid, orange juice, coffee, they will have a high level of hydrogens. Neutral pH is 7, so our pure, perfect water. Your blood should be around a neutral pH. And then base or a basic solution is 8 through 14, so that's going to have less of those hydrogen ions. And that's going to be our cleaning solutions, milk, toothpaste, stuff of that sort. So pH, a low pH or a high pH, it can destroy compounds, it can destroy our enzymes. So, for example, if you were to take acid, if you want to do an experiment at home, take lemon juice and add it to your milk. Those extra hydrogens will cause those milk proteins to crumble up, fall apart, clump together. That's literally the enzymes and the proteins denaturing. So cells have to have a specific pH and temperature so they can remain balanced. Luckily, in our bodies, we have these things called buffers, which help maintain the proper pH. So they'll add or remove hydrogen ions. So we have this in our blood in order to keep our blood pH at around a 7.5, I believe. So this is just a great example. So buffers, they're going to regulate the pH in our body. That's it for enzymes and buffers.